Hey everyone, so if you've been following this series of videos, I think you are ready to understand the project that we have been working on in the Hammond Lab as part of BSc 290, 299 undergraduate research for the fall, summer 2023 and fall 2023 semesters. So far we've covered sort of an introduction to Neurospora biology, where we covered the essential aspects of Neurospora biology, including mating, that we need to understand this project. We covered the discovery of the Neurospora spore killers, Sk1, Sk2, and Sk3, with an emphasis on Sk2 and Sk3. And we've covered the discovery of the Sk2 and Sk3 rsk gene. So the gene that Sk2 and Sk3 uses to resist spore killing, or to resist the effects of its own poison protein, of their own poison proteins, I should say. Now, what we don't know what the poison proteins are in Sk3. We have an idea of the poison protein in Sk2, but our project for summer 2023 and fall 2023 is to identify the poison genes, the genes that control poison protein production, assuming that poison is a protein. Our goal is to identify those genes in Sk3. And this work has been made possible by a study that was published from our laboratory, Velasquez et al. 2022 in Micro Publications. And so that article is about the discovery of a gene called a mutation called rfk-2 UV. And in this video, I hope to describe or teach you, if you're watching the video, of how we discovered this mutation and how our current project for summer 2023 and fall 2023 evolved from this discovery. So what Velasquez did, Velasquez et al., I should say, what they did, these are a group of undergraduates working in, I think, between 2019 and 2021, if I remember correctly. The publication didn't come out until 2022, but so they're working for a couple of years in the laboratory, and their goal was to isolate a mutant of Sk3k, so a spore killer 3 strain that kills spores by the Sk3 mechanism. So isolate a mutant that could no longer kill ascospores. Okay, and so they used a genetic screen to isolate this mutant. And this genetic screen is based on a similar one used with Sk2 that was published in 2014 by Harvey et al. So the same screen they used for Sk2 to identify the poison gene for Sk2, Velasquez et al. Is using it to discover the poison gene for Sk3, or at least they're going to try and use this same screen. So how does it work? How does the screen work? So what they did was they took crossing medium. So they used Neurospora crossing medium which many of you in the lab have already made and have already used for crossing. And they grew on the crossing medium. Let's say this is our crossing medium in a petri dish. And they grew a strain with the Sk3S genotype. It also has the fluffy minus mutation. So what that means is this strain cannot make macroconidia. So it's really convenient to use in crosses. So what they do is they inoculate this medium with this strain, Sk3S, with the fluffy minus mutation, and it grows across the medium, and over 6 to 10 days, it makes mycelia, but no macroconidia, and it also makes the protoparesthesia. So those are the protoparesthesia. Now, these will remain as protoparesthesia. They will not form mature fruiting bodies or the paresthesia until they are fertilized. And so how the screen works is the students generated lots of conidia. Let's say this is our typical culture tube containing slanted medium. And in that tube, they have a strain that has the Sk3k9151 delta HPH plus genotype. Now remember, if you remember from the previous video, this strain, when it's deleted of 9151, it no longer has an antidote protein, can no longer make an antidote protein to the poison that it makes. It can no longer neutralize the poison. So, and that's going to be important. Okay, so they generate lots of conidia, the orange macroconidia. And then what they do is they isolate all that conidia and they make a water suspension. This is not to scale. And they put that water suspension underneath a UV light. Okay, so here are the millions or billions of conidia that they have here. And they place it under UV light. And at intervals, various intervals, I think 15-second intervals, they take conidia from here, and it's been mutated by the UV light, and they drop the conidia onto the protoparesthesia of the Sk3S strain. So these are opposite mating types, and they're dropping the conidia on here, and the conidia have been mutated, and they're dropping the conidia on here, initiating fertilization of these protoparesthesia. And you can say, let's imagine each plate has tens of thousands of protoparesthesia, and maybe thousands of them get fertilized. So each crossing plate will have thousands of paresthesia being produced. Now, as we covered in previous videos, the paresthesia will mature, and each paresthesium in Neurospora crassa has on average 200 to 400 asci. And when the cross is Sk3k crossed to Sk3S, asci generally look like this: 4 viable ascospores and 4 dead ascospores. When the cross is Sk3k9151 delta, the antidote gene has been deleted, so there's no antidote anymore. The ascus phenotype is completely barren. So no ascospores, no offspring. And to see why that is the case, you should go back and watch the previous video where we talk about the killer neutralization model and how the killer neutralization model explains the absence of ascospores when there's no resistant protein around, when there's no protein to neutralize the poison. Okay, so we have that strain, Sk3k9151 delta, that's being mutated by UV light. So all the conidia here have this genotype, and they're being mutated by UV light. And we're initiating fertilization of the protoparesthesia, and as a result, they are forming paresthesia with 200 to 400 asci. And let's say there are approximately 10,000 genes in Neurospora. And so we can expect maybe 1 in 10,000 if we're lucky. Well, the calculations aren't very good. So it's a very rare occasion, but only when, well, let me say it this way. Most of the paresthesia are going to form empty asci, right? But let's say 1 in 100,000 or 1 in 1,000,000 times, one of the conidia will be mutated in a poison production gene. So this genotype right here, Sk3 killer, which no longer makes the antidote, if it can no longer make the poison because it has been mutated in a gene required to make the poison, and we're going to call that gene RFK, required for killing. So if the RFK gene is mutated, only then will we get a paresthesium that makes viable offspring. And so I'm just going to show 2 asci in here, and they're making the viable offspring. So every other paresthesium that's getting mutated by is getting fertilized by conidia that have been mutated by the UV light, but they've been mutated in genes that aren't required for poison production, and so they're resulting in barren asci with no ascospores. Okay, now remember, we didn't talk too much about this, but I did allude to it in a previous video, is that Neurospora crassa shoots the ascospores out of the paresthesium when they mature, when the ascospores are mature. And so right here I'm diagramming these ascospores flying out of the paresthesium at maturity. And usually if Neurospora is going to shoot anything from the other paresthesia, they're just going to be like dead ascospores or empty asci, just cytoplasmic junk. And these will the ascospores will tend to stick to the lid of the crossing plate. So what the undergraduate students did with this project, they made many of these plates, and looking for that rare occasion where you'd get some black ascospores stuck to the underside of the lid of the crossing plate. And that would suggest that, oh, okay, they successfully mutated a poison gene, because that's the only way we can get viable ascospores produced. Okay, so what they did next was, let's say this is the lid of one of their crossing plates. They would put it under a dissecting scope, and they would look for any black ascospores. And if they saw some black ascospores, what they would do is they would transfer them all to 500 microliters of sterile water in a microcentrifuge tube. So transfer all of them to the tube. Then they would heat shock those ascospores at 60 degrees Celsius for 30 minutes, because Neurospora crassa ascospores need a heat shock step in order to initiate or break their dormancy and get them to germinate on medium. Then they would plate those onto a medium suitable for germination and growth and formation of colonies from the ascospores. So let's say these are the ascospores. They spread onto a plate of medium, and they incubate for about 24 hours, and these ascospores start to form colonies. And then what they do is they look under the dissecting scope again and transfer each one to an individual tube, culture tube, containing growth medium. Now each of these tubes gets a name similar, based on the initials of the student who isolated the strain. So let's call these might be MAV1, MAV2, and so on and so forth. Or they could be MEW1, MEW2, the initials of students who worked on this project. And there were many other students, MEF1, MEF2. So M stands for mutant. And if you go to this paper, Velasquez et al. 2022, you look in the acknowledgments, you get the names of all the students that worked on this project. So at this point now, the students have to analyze each one to see if it's a true mutant, to see if it really is a strain with the genotype Sk3k 9151 delta HPH plus and RFK-UV, where RFK is required for killing and it is a candidate poison production gene. Okay, so how did they do that? So, working with each mutant, what they did, they first need to confirm that the genotype is Sk3k, right? Because they did it cross to Sk3s, and there's a chance that some of those offspring could have the Sk3s genotype. There is a chance that some of them could have the Sk3s genotype. So they need to confirm that the mutants, the putative mutants, have the Sk3k genotype. And remember, Sk3k is associated with a recombination block over a large part of chromosome 3. And the sequences within the recombination block are very different from the sequences within the similar region in Sk3s. So they confirm by PCR various locations and confirm that, okay, yes, the mutant is the Sk3k genotype, or at least in the Sk3k genetic background. So second step, they confirm that the Sk3k strain has the 9151 delta HPH plus allele, and it should have this. It should have this, unless we had some contamination and some other strain got into their crossing plate. It should have the 9151 delta HPH plus strain, assuming it is also Sk3k. So they confirm that by PCR and also hygromycin test, right? Because the strain should be resistant to hygromycin. And third, they confirm that the ascus phenotype is 8 black spores, 0 white. Okay, so remember, the original strain that they exposed, they collected conidia from the Sk3 strain, 9151 delta, replaced with the 9151, replaced with HPH plus. Now that strain, when crossed to Sk3s, should produce empty asci, right? Now, if they confirm these two and do the crossing test and they see this, it turns out that they didn't isolate a mutant. They isolated something else. They didn't isolate a strain that was mutated in a gene that was required for poison production. Okay, so most of what they isolated gave us this. Okay, that's upsetting. But once in a while, they did isolate some of their strains, I should say, had this genotype, Sk3k 9151 delta, or 9151 deleted with HPH, and were mutated in an RFK, a required for killing gene. And the phenotype was like this. 1, 2, 3, 4, 5, 6, 7, 8. So 8 viable ascospores. And presumably half are Sk3k, and the other half are Sk3s. Not just Sk3k, though, right? So 9151 is deleted, and the RFK, mutated RFK gene, is also associated with these 4 offspring over here. Okay, so once we focused in on one particular allele, one particular mutant allele, we named that one RFK2UV. So one of the mutations they isolated, we named RFK2UV. And what about RFK1? Well, RFK1 is a poison gene found in Sk2 strains, Sk2. So RFK1 has already been used up. So we named this one, this mutation that eliminates killing in Sk3, we named it RFK2UV. So now we need to find out where in the genome RFK2V is located. So turns out it's on chromosome 3, which is good because that's where we think all the important genes are for spore killing. That's where the resistance gene is located, and obviously we hope the poison production genes are found in the same location, and it was found on chromosome 3. Now, what Velasquez et al. Did was they used 3-point crossing to show if this is chromosome 3 in an Sk3 strain, chromosome 3, what they do is they use genetic markers. One is going to be called HPHA and one is going to be called HPHB to show that RFK2UV is somewhere over here to the left of HPHA. And we have a specific location. We can look at the paper. We know exactly where on chromosome 3 HPHA is located. And the 3-point crossing data say RFK2UV, whatever gene RFK2 is found somewhere to the left of HPHA. And this is a pretty long region right here. I think it's like a couple million base pairs long. So that's where that Velasquez et al. Study ended. And so now our next goal is to refine the locus. Refine the locus, just like we did for, let's say, remember RSK2 when we were hunting for the resistance gene, the gene that confers resistance to spore killing. I think in a previous video, we addressed that in a previous video. So we want to refine the location so we can identify where RFK2UV is located. And so what we did was we started putting additional genetic markers into chromosome 3 of an Sk3 strain. So we have Sk3 here. So it's just a standard Sk3 strain. And we put different genetic markers in. And what we're trying to do is identify if RFK2UV is, let's say, to interval 1 here, 2, 3, 4, or 5. So we're refining, narrowing down the locus of the location of RFK2UV. However, one of these genetic markers, so these markers we like to use, is the same hygromycin resistance gene. So we place it at various locations, one location at a time in an Sk3 strain. What we did was one of these markers, which we call vector 350. So when we replaced, when we inserted marker V350 into the Sk3 genome, something interesting happened. So let's say these are 2 genes that are predicted to exist in the Sk3 genome. One gene, the coding sequence goes from right to left. This gene over here, the coding sequence goes from left to right. Now what V350 or vector 350 is, it was designed to put a hygromycin selectable marker, HPH+, let's say, replacing these sequences here between these two genes with this hygromycin resistance gene right here. So replacing this with this. And what happened was when we did that, we have our Sk3k genotype with vector 350. We call the interval that's deleted by the hygromycin marker with vector 350. We call that the V350 interval. So the V350 interval deleted by HPH plus, when that strain is crossed to SKS, what happens? Killing is lost. So all the ascospores survive. Okay, so what does this mean? 1, 2, 3, 4, 5, 6, 7, 8. So half of these have the Sk3k, vector 350 delta, HPH plus genotype. The other half, they're all survivors, have the Sk3s genotype. So they all survive. So vector 350, what it did was it eliminated the ability of Sk3k to make the poison. And okay, so why? How is that possible? We just deleted some sequences upstream of this gene here and upstream of this gene here. So we don't know exactly why. At this point, we don't know why this has eliminated spore killing. This eliminates spore killing. It could be that somewhere in here is a hidden gene that is not possible currently to predict with algorithms that computers use to predict genes, such as this gene here and this gene here. There could be a hidden gene in there, and that hidden gene might be the poison production gene. It could be a regulatory element that controls expression of this gene here, or this gene here, or a gene at another location that is required for poison production. Another possibility, another thing we need to consider is this gene, is this the rfk-2 locus? Is this where rfk-2 is located? Right? So we put this marker in here to help us refine the location of rfk-2 by 3-point crossing. But it could be that this is where rfk-2 is located, and we actually found rfk-2. So we don't know that yet. Now, what you students, if you're watching this video and you're watching this video to help understand the project you've been working on this semester, what you're doing is you are deleting various segments of this interval. So we know, or we suspect, because we've done this one time, you're confirming this too, is that if you delete this interval here between these two genes, that Sk3 can no longer kill spores. So if you delete this region from an Sk3k strain, it can no longer kill spores. Each of you is deleting different regions. So one of you might be deleting just this segment here, another one might be deleting this segment here, and so on and so forth. Some of you are deleting longer segments, some of you are deleting this gene, some of you are deleting this gene. Some of you, I think, are deleting like the whole entire locus to see what happens. And we're going to combine all of your data to try and make sense of why this vector 350 based deletion eliminates spore killing by Sk.